Hello and welcome. Some 165 vaccines, according to the New York Times coronavirus tracker, are being developed around the world. 31 of these are in clinical trials and 2 are already approved for early or limited use. The trials range from preclinical trials, which involve animals, to phase 1 safety trials, to phase 2 expanded trials, to phase 3 efficacy trials, where thousands of people are tested before regulators in different countries approve. The research, of course, continues. The ones that have been approved are Chinese and Russian and called Ad5 and Sputnik 5. So to understand where we are in the vaccine journey and where we are headed and how you should be looking at potential vaccines or for that matter cures in the market, I'm now joined by Dr. Gagandeep Kang, clinician and scientist who built the national rotavirus and typhoid surveillance networks as well as laboratories for clinical trials in vaccines. She is a professor at the Christian Medical College in Vellore, where she's returned after several other stints, including with the government at the Indian Council of Medical Research. Dr. Kang, thank you very much for joining us. So the straight question first, where are we from your perspective and vantage point in the search for a vaccine for COVID-19?
If we look globally at what's happening in vaccines, unprecedented pace of development, and one of doing so quickly is because we have learnt from Ebola. In the case of Ebola, we had a vaccine that was sitting in a freezer, but it took us 9 months to get all the paperwork in place to begin the evaluation of the vaccine. After that, the world got together and said we weren't going to let that happen again. And that is the reason why within 7 months of recognizing that there was an outbreak, we have 30 vaccines that are in clinical trials already. This is fabulous for science. It's also hugely challenging because we are directing a lot of resources towards vaccine development and not necessarily in other measures of control. That is the global scenario, and where we are in India, we also have a lot of vaccine candidates that are being developed by our companies. Many of these are in partnership with global players. Some of them are being developed indigenously as well.
Right. So, and that's an interesting point, Dr. Kang. You're saying that we are devoting a lot of resources to vaccine development and not enough to control. So, could you dwell on that a little more, and why is it either or?
It isn't really either or. In an ideal world, you would have enough resources to direct towards all the public health strategies that are very important, which is testing, tracing, and isolating. You would have enough resource to treat people who got sick. You would be looking at developing new and easy diagnostics. You would be looking at repurposing drugs as well as designing and developing new drugs. And you would be focusing on prevention through vaccines. For reasons that are not completely understood, it almost seems like the world thinks that vaccines are going to be a panacea for everything. Once we have a vaccine, the problem is solved. Well, while I'm a huge proponent of vaccines, I know vaccines are only one part of our ability to handle infectious diseases. So the focus on vaccines is good, but there's a lot more we need to do.
Right. So then let me come back to vaccines in a moment. So the other element in the equation is obviously a cure. So does the cure now appear conceptually also so distant that maybe we are putting all our eggs in the vaccine basket, so to speak?
I think a cure means an antiviral. And we have to remember that in this case, the disease progresses and needs different kinds of drugs at different stages. In the early stages, certainly an antiviral would be a good idea. But much of the morbidity and the mortality that we are seeing because of COVID-19 is not anymore because of the virus. It really is because the immune system has taken over and is dysregulated. In trying to eliminate the virus, it overshoots and results in multiple consequences that then become difficult to arrest. So, understanding the virus and understanding the disease process in people who are severely ill is very important.
And that's an important distinction. So, in your mind, what would the cure or the components of the cure be as they are being worked upon right now?
In early disease, we would be looking for an antiviral so that you don't go
Right.
on to severe disease. And once you are in severe disease, you will be looking for which are the pathways that are impacted by viral infection and what can you do about that. So, for example, remdesivir is an antiviral, and we should be evaluating whether we should be using it earlier and earlier in illness. And dexamethasone is a steroid, and its role really is in severe disease. So, whatever the course of infection we are looking at, we need to be looking at evaluating different approaches for modulating both mild and severe illness.
Right. So, from what we know about this disease, it is fatal, and or it can be fatal. Not, in some cases, of course, not in all cases. But our understanding of the medicines that are being administered is that for who it, I mean, for those it works, and for others it doesn't. And I think the way you've explained it, in situations where, let's say, it crosses a certain tipping point within the body, and the immune system begins to fight back, then no medicine really works. So is that then, as you yourself pointed out, something that goes beyond COVID itself and is akin to maybe some other disease conditions which we've either known or not known?
So, yes, there are multiple components that you need to look at. One is the effect on the lung, where we know that the lungs in COVID-19 disease are not like the lungs that you see in other kinds of pneumonias. The lungs here, the happy hypoxia that people describe, that your oxygen saturation drops without necessarily a person feeling uncomfortable or breathless, seems to be something that we have not seen for other diseases. And one of the treatments that we have found for that is something that was known before but was not applied as widely. And that is the ability to increase oxygenation of the lung by having people treated on their stomachs rather than on their backs. That works just with room air, and it works when someone is being administered additional oxygen through a nasal cannula or a mask. So these approaches help. There is the lung aeration, which is important. The second component to look at is the suppression of inflammation, which is what dexamethasone does. And the third component to look at is what's happening with the blood vessels, because that is what results in impact on multiple organ systems in the body, not just the lungs. We now know that low molecular weight heparin can help. So anticoagulation and the point at which anticoagulation should be used are things that have been tested and tried only in the last few months. I'm hoping that there will be more such experimentation which will lead to better patient survival.
Right. So you mentioned Ebola in the beginning. So when you compare Ebola and other diseases that you've encountered or worked with coronavirus, what in your mind are the two or three things that make it fundamentally different, and therefore everything that we see around us?
So from the public health point of view, a lot of what you need to understand about diseases is what's the transmissibility of the disease, who gets it from whom and how. In Ebola, we knew very clearly that Ebola was person-to-person transmission. You needed to be in contact with the patient to acquire Ebola. So therefore, the reproductive number for Ebola was relatively low. And that's why when we saw an outbreak, the largest outbreak we have ever seen was 30,000 cases. In the case of SARS coronavirus 2, what we are seeing is a virus that spreads through the respiratory route, which means you don't need to be in direct contact with the person to acquire the infection. And the other big difference is that it spreads when a person is asymptomatic or pre-symptomatic. And that is what is resulting in the high rates of infections that we are seeing here. So, from a public health point of view, from the view of what kinds of control measures we need to be using, these are two very different disease conditions. Then you
Right.
come to the clinical picture and others, and of course, every disease is different in that context.
Right, and I'm going to come to the control part in a moment again. Now, on back to vaccines. So, given what we know today, what's your sense, particularly if you were to explain to a layperson, of the efficacy of the vaccines that we are seeing rolled out? I mean, even if you were to ignore the ones that have already arrived, let's say the Oxford vaccine, which may come at the end of the year, and what is the likelihood that they could be a solution to everyone or for everyone?
So, we actually have absolutely no knowledge of the efficacy of any vaccine at this time. That's why we do phase 3 studies, the efficacy studies, to see whether vaccinated people develop less disease than people who are not vaccinated. We are now in multiple phase 3 trials with multiple products, and we need to wait for the interim and final results from those trials, and then we'll have answers. But given that this is a virus about which we have learnt a lot, given the data that we have from some monkey studies, I think there is a very high likelihood that we will have a successful vaccine in the next 6 to 12 months. And it's important to remember that having a successful vaccine is only the first step. These vaccines are tried first in young and healthy people. The people who are most affected are the elderly. Will this vaccine work in the elderly? We don't know. The Oxford trial does include a small number of the elderly, but it will not be sufficient to give us efficacy data. We will get data on the immune response in the elderly from that trial.
Right. And what will a vaccine do? I mean, again, this is a layperson's question, doctor. So, what will a vaccine do, and how will it work in the case of coronavirus, and how will it work if it works?
If we were to look at what an ideal vaccine
Yeah.
was, it should be a vaccine where I take one shot. Preferably, it should be a nasal spray so I don't even have to take the shot. And I am protected from infection for life. I never need to take another booster. I can go out safely. I know that I will never acquire this infection. Unfortunately, those kinds of perfect vaccines are very rare. We don't have them for practically any disease that you care to name. You do need multiple injections. You might require boosters. It may not work for everybody, certainly wouldn't work in people who are immunocompromised. So, if you look at what a vaccine does, one way of looking at how we might expect the vaccine to perform is to look at influenza. In the case of influenza vaccines, we have to make a new vaccine every year because the strains change. We need to take the vaccine every year because protection is short lived. And there is some level of memory, but it isn't huge. So, in some years, the vaccine might not work at all. We are not going to see that kind of variation with the coronaviruses. I think we will have an immune response, but we don't know the duration for which people will be protected and whether that protection will be from severe disease or from infection. Lots of people get infected, not everybody gets disease. So, our low bar is this must prevent disease. And if we look at what the FDA, which is the US regulator, and WHO have recommended, they have said that they will license vaccines that have 50% efficacy as a point estimate, which means the vaccine in the trial should prevent at least 50% of confirmed disease. This is not about infection.
Right, and if the vaccine is has that level of efficacy to the extent that you're allowing for it, will it prevent any other diseases as well, infection from other diseases as well, or is it sharply focused on preventing SARS coronavirus? If it has that level of efficacy and applicability.
So all vaccines have a target, and all SARS-CoV-2 vaccines have a target, which is SARS-CoV-2. Now, if we look at influenza vaccines prevent influenza infection. But if you don't get an influenza infection for the elderly, that's actually a good thing, because then you don't get a secondary bacterial infection, which might be the thing that makes you really, really sick. So the vaccine is not intended to prevent that secondary bacterial pneumonia, but it does by making sure that you don't get the viral infection that precedes the bacterial pneumonia. Since this is a respiratory infection, we might see something similar, that we would not get an infection, a secondary infection. We don't know the extent to which secondary infections are the killers in SARS-CoV-2, but it's early days yet for that.
Right. Dr. Kang, we've seen many of these sero surveys for Mumbai. We've just seen it for Pune, where it's talking about a 50% antibody presence, Delhi. What is this broadly telling you?
It's telling us about what proportion of people who are in the areas that were sampled have been exposed to infection. So the presence of antibodies means that you were infected at some point in the past and your body has responded to that. So for one, it tells us that in these areas, just counting the cases was insufficient. It can tell us by what factor we were off in the counting of cases. And in the future, when we have a better understanding of what immune responses really mean, it might tell us how long we might be protected from infection. Those are not answers we have yet, but serosurveys will be very useful for us to continue to track infection and the potential for populations to be protected from subsequent infections.
But is it giving you comfort, or is there in any way that more people than perhaps at least laypersons like me thought have actually contracted the virus and not been affected by it?
Yes, it does. So one of the ways to think about it is actually in Mumbai and in Pune, the populations that were selected were chosen because there were high rates of disease or intermediate levels of cases being reported from those locations. And then we find that we expected maybe 10% of the population to be affected, and it's actually 50%. So it is a good thing in that it tells us that we might be actually closer to herd immunity than we thought. But you could also look at it as a negative to see, okay, what else is out there that we missed that we should try to have a better handle on in the future.
Right, and that comes back to the point that you made earlier. So how do what according to you should be our focus and thrust in terms of management of this disease, including the controlling, prevention of further transmission, and testing, and so on?
I think the principles stay exactly the same as they were right at the beginning. You should have the opportunity to test as widely and as frequently as you can, and act on the results of those tests rapidly. It's not useful when you do a test today and you get a result 3 days or 5 days or a week from now. and it's not useful if you don't have the ability to act on it. What you're trying to do is to prevent transmission from an infected person so that the overall numbers in the community come down. That can only happen if we test and we make sure that we isolate quickly. If there have been contacts, obviously they should be traced and tested as well so that we can bring down the circulation of the virus in the community. We've seen from examples around the world that this is feasible, but it does require a lot of resources. So we have to balance what is practical with what is ideal.
And finally, Dr. Kang, as you look ahead, we are still, in terms of number of cases, the cases are still rising in India. India was much behind earlier. Now it's above. It could be to do with the amount of testing we're doing. What's your sense? Is it? Are you getting a feeling that things might start plateauing and going down in maybe the next 3-4 months? Or in any case, I'm sure you would advocate for alertness, but and therefore how alert should we be and till when?
So I think one of the things to remember is that India is a very, very, very large country in terms of its population. We also have a range of population density that we live with right now. What we've seen is that the most densely populated areas were infected first. Now we are at tier 2 cities getting infected. We still have a long way to go. The cumulative numbers, I don't think the patterns are going to change. Where the patterns play out will change as the disease spreads to different parts of the country. So it's very difficult to say that we are plateauing. We are not only plateauing, we are heading downwards in some places, but we are on the upswing in others.
Right. Dr. Kang, thank you so much for your time and sharing your thoughts. And I do wish you and your colleagues all the very best in this continuing battle against coronavirus. Thank you so much.